I am Edward Dennis, and I welcome you to this lecture on bioenergetics and mitochondrial functioning. This lecture will be in two parts. First, we'll focus on bioenergetics, including the several forms of biological energy, how they're translated into electron transport gradients, and we'll conclude talking about the poisoning of gradients with several examples of gases that function to inhibit the electron transport chain. In the second part, we will turn our focus to mitochondria, which is where all of these processes occur, and first explore the chemiosmotic hypothesis and how that works to drive ATP synthesis. Then we'll spend the majority of the time with an overview of mitochondrial diseases, for which understanding the underlying bioenergetics is essential in order to understand and evaluate mitochondrial diseases. The learning objectives for the subject of bioenergetics and mitochondrial functioning are listed here, and I urge you to look these over before starting the lecture. The reading assignment for this lecture is listed here. I want to start this lecture with a clinical problem. You are a neurologist, and you are asked to see a 13-year-old girl who has a 2-year history of a variety of symptoms, including periods of limpness or twitching in her extremities, sometimes with blindness, hearing loss, an awkward balance and gait, aphasia, and memory loss and seizures. In an MRI scan of her brain, there are some focal areas, as illustrated. And you are stuck with the question of what do you tell her parents and what is her disease. This is an unusual disease, and in order to appreciate its ramifications and what causes it, we really must first look at some underlying biochemistry and biophysics of electron transport. And then at the end of Part B, we will come back to this case as an example. Let's first summarize some earlier lectures in which the point was made that everything we eat breaks down to acetyl-CoA, whether it's proteins or carbohydrates or lipids. The former two first are broken down to amino acids and simple sugars, and whether they go through pyruvate or not, and along with fatty acids, one ends up with acetyl-CoA that then goes through the TCA cycle, generating units of NADH and FADH2. And these products then are converted into energy forms in the form of ATP. And this occurs through a proton gradient that makes use of some complicated molecules called cytochromes. There are three important forms of energy and energy storage in biology. They're listed here. The first is in the form of phosphate bonds that are incorporated into ADP and ATP, and these store chemical potential. A second form is in the form of electron carriers, such as NAD and ADH, and these have electronic potential. Finally, we come to the third form, or gradients, usually proton gradients, and this has a form of chemiosmotic energy that is what is used in the mitochondrial matrix. And these forms are constantly being interconverted during metabolism. Perhaps the simplest form, and the one you're most familiar with, is ATP, which stores its energy as phosphate bonds. So ATP, as you know, is composed of an adenine base on a ribose background, which you've heard a lot about in earlier lectures. And together, the ribose and the adenine make adenosine. When there is a phosphate on the adenosine, then it is adenosine monophosphate, diphosphate, or triphosphate. And it is the high-energy phosphate bonds, the pyrophosphate bonds, of those second two phosphates that is where the energy is stored. The two major electron carriers are NAD and FAD. If we look first on the left at NAD, NAD has a positive charge, and when an electron plus an H plus, together known as a hydride ion, a hydrogen with two electrons, which has a net negative charge, is added to the positively charged NAD+, one obtains NADH. In an analogous fashion, if one takes FAD, shown in the upper right, and adds a hydrogen with an electron, a so-called H dot, it adds to the NAD and results in a radical or semiquinone form. And when a second H dot is added, that combines with this form to make FADH2. The net result is that two hydrogens have been added, two H dots. That would be the equivalent of an H plus and an H minus. And we go from a starting neutral molecule to a resulting neutral FADH2, which is the reduced form. In the case of NAD, we start with NAD plus and add a hydride, and we end up with NADH, also a neutral form. There is another form of electron carriers that is very important here, and that's called coenzyme Q. Coenzyme Q, shown in the upper left, is also a neutral molecule. It has a number of isoprene units attached to it, a subject that we discussed in the process of talking about cholesterol synthesis. Coenzyme Q picks up an H dot to make coenzyme QH, as shown in the middle, and picks up a second H dot to make coenzyme QH2 or ubiquinol. You'll notice that it went from a neutral molecule to still a neutral molecule, having picked up two H dots, or again the equivalent of an H plus and a electron. As we will see, coenzyme Q plays a special role in transporting electrons within membranes between complexes of the electron transport chain. Now we come to the third form of energy in the form of gradients, and this is illustrated with the classic electron transport chain. It is located in the mitochondria on the inner mitochondria membrane portrayed at the top, and we'll say more about the details of this subsequently. The important point here is that there are several complexes in the electron transport chain, numbered 1 through 4, and the NAD is generated from the inner part of the mitochondria, and that NAD plus becomes NADH when it picks up an electron. That electron is then transferred to another receiver known as FMN, and eventually to coenzyme Q that I introduced on the preceding slide. Analogously, with some of the reactions, like the conversion of succinate to fumarate in the TCA cycle, FAD is reduced to FADH2 by picking up an electron, and that electron is transferred to coenzyme Q similarly to the preceding example. These electrons are then transferred to complex 3, and another protein known as Cytochrome C plays a similar role of taking an electron and transferring it to complex 4, but more about that later. Let's now look at the architecture of the mitochondria. In the center is the matrix, and the matrix is where the TCA cycle enzymes, fatty acid oxidation enzymes, and mitochondrial DNA and mitochondrial RNA are located, as well as the mitochondrial ribosomes. Surrounding the matrix is the inner mitochondrial membrane, and this is where the electron transport assembly is located that I discussed on the previous slide, including these complexes. The NAD and FMN is delivered to the electron transport complexes from the TCA cycle enzymes in the matrix. There is also an outer mitochondrial membrane, and the space in between the inner and outer is referred to as the inner membrane space. There's one more complex, and that is the ATP synthase, which is located in the inner mitochondrial membrane, and we shall discuss that in Part B of this lecture. With that as an introduction, let's look at the same features in terms of mitochondrial anatomy. Here is a mitochondria, and in the matrix or center of the mitochondria are the TCA cycle enzymes, the fatty acid oxidation enzymes. There's also specific mitochondrial DNA and mitochondrial RNA and mitochondrial ribosomes. There is an inner mitochondrial membrane surrounding the matrix, and this is impermeable to most small ions, as well as all sizes of regular chemical molecules. There's a intermembrane space outside of the inner mitochondrial membrane, but inside of the outer membrane. The outer membrane is more permeable to ions than the inner mitochondrial membrane. And positioned in the inner mitochondrial membrane is the electron transport assembly, which is where that NAD and FMN go to interact with the complexes I introduced on the preceding slide. There's one more feature here, and that is ATP synthase, which we will be talking more explicitly about in Part B of the lecture. Let's pause here and introduce two principles of proton gradients. The first is that energy transducing membranes, like the inner mitochondrial membrane of mitochondria that I introduced on the preceding slide, are impermeable to protons unless there's a pump or a carrier present. The second principle is that there is such a pump or carrier in the form of cytochromes that are arranged vectorially in the inner mitochondrial membrane of mitochondria. And they are able to transport protons through that membrane. One more fundamental point, and that is I want to introduce some nomenclature. The first is that of a porphyrin. A porphyrin is an organic molecule. It's illustrated here. It contains 4 rings. They're 5-membered rings, each with a nitrogen in the center. And this is a covalently bound complex planar molecule that can exist. When a metal ion is present, such as iron, it can be coordinated to those 4 nitrogens as well as to other groups because there are coordination sites both above and below the plane of the ring. When a porphyrin has an iron in it, then it's known as a heme. There are numerous porphyrin structures and numerous heme structures. Shown here are just 2 examples. On the left is heme B, and its formal name is iron protoporphyrin 9. If you look at its structure, it has the basic ring structure, and in red are several side chains. Now, this same protoporphyrin, when covalently bound to a protein, is called heme C. And you can see that 2 cysteines through a sulfur linkage is bound to the side chains at 2 positions on the heme backbone. So heme structures are non-protein prosthetic groups on proteins, and they can be either free and just associated non-covalently or covalently attached through proteins as illustrated here. There are numerous cytochromes. Illustrated here is just one particular one of the cytochrome B family, also known as complex 3. And this is composed of numerous subunits, numerous proteins, and it includes at least one with a heme group as the prosthetic group. This sort of cytochrome is typical of the cytochrome P450 class of enzymes, very important, involved in drug metabolism. The mitochondrial electron transport chains have cytochromes in them, and hemoglobin and myoglobin are composed of cytochromes. Let's quickly examine one other aspect of the mitochondria. And shown in the upper left is a blow-up of the matrix of the mitochondria, the inner membrane, and the outer membrane. And here I want to introduce cytochrome C, another form of cytochrome. It's a protein that floats around in the space between the inner and outer mitochondrial membrane, but associates with the inner mitochondrial membrane. When cells undergo apoptosis, then many pathways are affected, including increased membrane permeability of the outer membrane. And this leads to the activation of caspases and many downstream events consistent with and leading to apoptosis. Now, with all of these definitions in place, we can focus on the functioning of the proton gradient. Shown here are three of the key complexes, complex 1, 3, and 4. And they're positioned in the inner mitochondrial membrane in an oriented fashion. So the process starts with the generation of NADH, which passes its electron to FMN and through some iron sulfide complexes further processes that electron through complex 1, eventually delivering the energy to coenzyme Q, which is the mobile electron carrier shown here as positioned in that membrane and carrying the electron from complex 1 and delivering it to complex 3, where it goes through another number of complexes, eventually delivering the energy to cytochrome C, introduced on the last slide, which is associated with the inner membrane space side of the inner mitochondrial membrane rather than the matrix side, and is also a mobile electron carrier because it can pass the electron to complex 4, which is the end point of the electron transport chain. In this process of the electron going through these complexes, it generates energy in the form of a proton gradient. And you can see the three arrows. The number beside them should not be taken too literally, but the proportion of energy that is resultant in a gradient from complex 1 and coenzyme Q is each about double that from its final product in complex 4. I want to now conclude this lecture by illustrating three different gas exposures that lead to poisoning of the electron transport chain. In the first example, we're talking about hydrogen sulfide. And you could imagine yourself being an ER doctor. A miner arrives by ambulance, and he is delirious. The paramedics say that he collapsed in his mine and later vomited in the ambulance. On examination, you see that his respiration is slow, his lips are bluish, they use the word cyanotic, and a chest x-ray is normal, but he smells like rotten eggs. And while you are examining him, he has a seizure. So hydrogen sulfide poisoning is very uncommon, but when it occurs, it's typically among minors, and the symptoms of cell hypoxia, delirium, seizures, nausea, vomiting occur, and just because it's hydrogen sulfide, it has a particular smell. What is it doing? It's stopping the electron transport chain. And treatments include supplemental oxygen, hyperbaric in particular. Various nitrites can reverse the effects. A second example is cyanide poisoning. Again, it's a very rare occurrence. Usually it's caused by industrial chemicals. Again, cell hypoxia, the similar symptoms to those experienced with hydrogen sulfide, delirium. In this case, there's a telltale smell on the breath of a bitter almond, characteristic of cyanide. And its mechanism is similar to hydrogen sulfide. It is inhibiting the electron transport chain, in particular cytochrome oxidase. And the treatments are very similar to that for hydrogen sulfide. The third example is carbon monoxide. Again, it's rare, but miners, firefighters, others can suffer from it. Again, similar symptoms, cell hypoxia, delirium. The mechanism here is twofold. One is it inhibits cytochrome oxidase, as do the other two gases, but in addition, hemoglobin can bind carbon monoxide and inhibit it, as is illustrated on the graph on the left. Again, supplemental hyperbaric oxygen can reverse it. To conclude Part A of these lectures, I've summarized here the three poisons that we examined. All three of them interact with cytochrome oxidase, another name for complex 4 at the end of the electron transport chain. They block further electron transport by so binding. And they all exhibit classic symptoms of delirium with slightly different parameters: seizures, a smell of rotten eggs for hydrogen sulfide, tachycardia, and a bitter almond taste for cyanide, headaches, cherry lips for carbon monoxide. And the treatments are similar for all three of them. The addition is that carbon monoxide also binds to hemoglobin and affects its functioning.